Welcome everyone. This week we're having a look at DNA profiling and we're going to answer the question, how is your genetic code translated into a protein? So, how is the DNA code translated into molecules called proteins, and how can this information be used in a forensic investigation are your learning intentions for today. And what we'll hopefully be able to get out of this is we'll be able to name the steps from getting from a DNA molecule to a protein molecule, and also be able to decode a strand of mRNA from a DNA sequence. And we haven't looked at mRNA yet, but that's what we're going to learn today. So what is DNA profiling? It's a process or technique that reveals the unique pattern of an individual's DNA. So this is the order of the nucleotide sequence, the bases, and how they're added together. So what order these A's, T's, G's, and C's are in. And so this process has been the most important discovery in forensic science to date, and it was discovered in 1984 by Alex Jeffries, who discovered this technique, which we call genetic fingerprinting, so working out the order of those nucleotide sequences. And in 1987, it was first used to convict a criminal in a crime. Yeah. So this is Alex Jeffries here in the lab, 1989. So what do we know about DNA already? So we know it's a double helix structure. We know that it's a polymer that's made up of monomers called nucleotides, and each nucleotide has a sugar and a phosphate backbone with a variable nitrogen base. So this can change, and it can either be A, T, C, or G. And so the sequence of how these nucleotides join together is the code for the protein, so the order of these nucleotide bases. So how is DNA organized? So we've seen this double helix structure of DNA. How is this organized inside the nucleus of our cells? So the long strands are called chromosomes, and what they are a condensed form of DNA. So we see our strand of DNA here. It wraps around these little proteins. These proteins stick together, and they are all bundled together as well, and they form this really nice, neat, compact structure. It's very condensed, called a chromosome. So we have a pair of 23 of these chromosomes, which gives us 46 in total. We get half from each of our parents, and the same thing for dogs, but they just have 38 pairs. So what is a gene? So a gene is a short section of DNA that codes for a protein. So it's a short section just like this that codes for a specific protein. And the sequence of the bases act as the code. So we have two strands here in our piece of DNA. We know that we have that double helix. Well, one of these is actually a coding strand, and one of these is a template strand, which is what I want you to really remember. We have a coding and a template strand, and I've spelt that wrong. Template. So the order of these bases of the coding strand are what instruct your cell to make a protein. So we can see that we have an adenine here, we have a cytosine here, we have another adenine here, we have a guanine here, and this template strand will be complementary. So we can see that that's a thymine, we can see that that's a guanine, we can see that that's a thymine, and so on. So the sequence of these nucleotides in the coding strand are what make your DNA, your proteins. They tell your body to make proteins. So this template strand is called a template strand for a reason, and we'll find that out shortly. So pause here and fill out questions 1 to 7 if you haven't already. So how do we get from a piece of DNA into a protein? So to make a specific protein, we need to make a copy of the gene first. So we can't just take this whole piece of DNA and make a protein from it, otherwise it'll be massively long, it'll be huge. So we need to make a specific copy of the exact protein we want, and that copy that we make of the DNA is called mRNA. So mRNA has almost the exact same structure as DNA, except it's not got that double helix structure, it's just a single strand, and it's made up of nucleotides as well. And it's just much smaller because it's only a single gene, whereas DNA has thousands and thousands of genes in it. So what is mRNA? mRNA is called messenger ribonucleic acid. So it's a type of RNA, so it's a type of ribonucleic acid. Another type of these is tRNA, but we're going to talk about that next week. But for this week, just remember that mRNA is a type of ribonucleic acid. It's a messenger. So what it is just a photocopy of a gene. So it's very, very similar to DNA, except it has a few little differences. The first difference is DNA has this deoxyribose sugar in it, whereas RNA has a ribose sugar, so it doesn't have that deoxyribose, it's just got a ribose, which is why it's RNA and not DNA. And so if we have an exact copy of a gene of RNA, we have that nucleotide sequence here, and that's the copy of this coding piece of DNA. The sequence is the exact same, except RNA has a uracil base instead of a thymine. So this just replaces the thymine. So if you're ever doing a question where you had to write the complementary RNA strand that's being made, instead of putting in a T in complementary to an A, we will put a U in place of that T. And one thing to remember is it's single-stranded, whereas DNA is double-stranded as well. So how is the mRNA made from DNA? So DNA is too big to leave the cell, so an mRNA copy of the gene is made. So the DNA strand separates, and then mRNA, the messenger ribonucleic acid, is built using the DNA as a template. So that's what that template stands for. So we can see if we look at this diagram, we've got this piece of DNA, and we have the coding strand and the template strand. So the coding strand has the information for what the protein is, and the template strand is complementary to that. So the mRNA molecule will be built off the template strand, and you can see that if we do the exact same thing, we add these complementary bases. So instead of having a T here, we now put a U because RNA has a U instead. If we keep on adding these complementary bases, what happens is this RNA strand is the exact same as the coding strand, except for that replacement of uracil with thymine with uracil. So this process of building this RNA molecule off a template strand is called transcription. So that's the one word I really want you to remember today is transcription. So building that mRNA strand off a template strand is called transcription. And it's just the complementary bases, similar to if we were doing questions where we make the complementary DNA sequence. It's the exact same, except we just add this uracil instead of a thymine. And that gives us our transcript at the end. So we have our transcript of the DNA, which is our mRNA molecule. And it's the exact same as that coding strand, except for uracil. So what actually drives the transcription of mRNA? So we can obviously do it in a question. We could see that we could write in the complementary bases if we really wanted to. We can work out what that code is. But how does your body actually do that? So there's this little machine in there, and it's called RNA polymerase. So what it does is it attaches the mRNA to the DNA strand it attaches. What happens first is that the RNA polymerase, this big molecule here, will attach to the DNA in this specific region. This is called the promoter region. So it's the promoter region. And basically what it does is it just plops along here and it unwinds the piece of DNA and opens it up like this. So it's just opened it up and it's accessing this template strand here. And like we would do it, where we know the complementary bases to add, well, RNA polymerase also knows this, and it moves along and adds them to that template strand and results in this nice piece of mRNA being formed. So RNA polymerase is what's responsible for unwinding the DNA and accessing that template strand. And it also reads the template strand and constructs the chain of the complementary nucleotides. So it adds the A's and the U's and the C's and the G's. So a specific sequence of nucleotides will be found at the end of a gene, and that'll tell the RNA polymerase to stop what it's doing and let go of this mRNA molecule so it can go off to be used to make a protein. And so that's called the termination site, sorry. So this process is called transcription. Let's remember that. This is transcription, and RNA polymerase is what drives transcription. So how do we get from a strand of DNA to a protein? This is a great video that illustrates it, and you'll see this piece of RNA polymerase moving along the gene and adding on these complementary nucleotide bases to the template strand, which will end up having the exact same order as the coding strand. So I'll leave the link for that video on your worksheet and have a watch from 0 to 1:15. So in summary, guys, the process of synthesizing a strand of mRNA from DNA is called transcription. Remember that. RNA polymerase is the key enzyme that is required for mRNA construction, because that's what comes in and unwinds the DNA and adds the bases. So mRNA is similar in structure to DNA, but it contains a ribose sugar instead of a deoxyribose sugar, and replaces thymine with uracil. So instead of there being T's, there's now U's in RNA. And lastly, your DNA code is unique to you unless you're an identical twin. But a DNA profile can be made from your genetic sequence and matched directly to you from a sample, which is why it was the most and has been the most important breakthrough in forensic science, being able to accurately match an evidence sample to a suspect sample. So remember, guys, we've looked at transcription this week, and that is the process of getting from DNA to mRNA. That is transcription. So what we're looking at next week is how that then turns into a protein. But for now, we're just remembering this step here is called transcription. That's the most important thing to get out of today, and the few steps that happen to get it. So have we got questions, the 8 to 13, finish it off, and have we got the rest of the activities for the week?